After discussing the principle of isothermal titration calorimetry and differential scanning calorimetry, we have now started discussing the applications of both these calorimetric techniques in academics and in industry. Today, we will talk about the use of isothermal titration calorimetry in rational drug design. We will discuss how the changes in enthalpy, entropy and free energy can be connected with the efforts to improve the existing molecules or new molecules with a focus towards novel drug design. There are a lot of synthetic chemists, molecular biologists and many other scientists working towards synthesizing, designing and coming up with new drug molecules. Every molecule which has to act as a drug molecule has a specific function, and sometimes the function can be it has to go and inhibit some process. For example, even if it is inhibiting the activity of an enzyme, then inhibition of that particular activity will require a specific binding of that molecule or of that inhibitor with the enzyme. And even if it is to be connected with the drug delivery, again the focus comes on the particular affinity of that molecule towards that protein or towards that enzyme. That means one of the efforts has been to improve the affinity either in the existing drug molecule or to come up with new molecules which should have improved affinity, higher affinity, affinity of the order of the desired target. We will discuss how isothermal titration calorimetry has given some guidelines over a period of years in this direction. Let us remember that the guidelines towards something do not come overnight. One has to work with a variety of molecules. Suppose even if the base of a molecule is same and then different substituents are added, the effect of different substituents on the binding affinity can be checked. Thus, by doing experiments with a variety of drug molecules, structure property activity relations can be drawn and some guidelines can be drawn for rational drug design and target oriented synthesis. Now, as I mentioned that there are large number of libraries of compounds available. There are new molecular entities being added every year, every month to this huge library of compounds. But each compound synthesized with an idea of making it as a drug does not generally end up in being a drug. What are the required properties in a drug? It should have a good oral bioavailability. It should have good solubility, it should have good membrane permeability amongst others. Now, for a drug to be orally bioavailable, the molecule has to pass some stringent requirements. And what are those requirements? Let us take a look at those requirements and let us go to the slide. Those requirements have to be met first of all in the form of Lipinski rule of 5, which is also sometimes called RO5. What is that rule? As I commented here, optimization means even if you are working on an existing molecule, you want to optimize the molecule so that it becomes an effective drug. And that is what is commented over here, that optimization needs to follow very stringent constraints to maintain drug-like character. And what are those? Suppose we want to optimize a molecule or come up with a new molecule which should be accepted as a drug-like character. And as I said, for a molecule to be for a molecule to have good bioavailability, the stringent constraints are, let us take a look at molecular weight should be less than 500, there should be no more than 5 hydrogen bond donors, there should be no more than 10 hydrogen bond acceptors, and there should not be any toxic group, and finally C log P should be less than 5. So, these 5 constraints 1, 2, 3, 4, 5, that is why it is called RO5, Lipinski rule of 5. So, in addition to the molecular weight and the hydrogen bonding abilities in the form of hydrogen bond donor or hydrogen bond acceptor, toxicity also becomes a very stringent requirement. Toxicity comes later, but before that C log P should be less than 1. Now, this log P, P is partition coefficient. C log P or log P actually tells about the hydrophobicity of a molecule, because P is partitioning coefficient between octanol and water. Octanol has one hydroxyl group and there are 8 carbons. That means it is a largely hydrophobic molecule. And when we have a mixture of octanol and water and you add a hydrophobic moiety to this, it will partition between octanol and water. So, if the molecule is largely hydrophobic, it will partition more into octanol, and if the molecule is largely hydrophilic, it will partition more into water. So, therefore, if there is more partitioning into octanol, that means the molecule is more hydrophobic. And the requirement is, let us take a look at this slide, that log P or C log P should be less than 5. That means there is a maximum limit set for the hydrophobicity of the molecule, and the effort has to be that P or log P should be less than 5. Now, let us take a look at this slide. Prevalent approaches have resulted into more hydrophobic compounds. Once again, commenting upon the synthesis of new molecular entities with an objective of rational drug design or novel drug design. We generally come across the information from the literature that most of the approaches in this direction result into more hydrophobic compounds. But then the question is, are these hydrophobic compounds or the compounds in which the hydrophobic component is more, is it good or is it bad for a molecule to act like a drug? And as I just mentioned, C log P should be less than 5. So, there it refers to that the hydrophobicity should not exceed a certain value. And I will talk a little more about it, that what the literature has suggested, whether the hydrophobic content should be more or polarity should be more. Now, let us take a look at the next comment. High quality compounds require high solubility, low log P and not invoking hydrophobicity. Now, this information is based upon the literature information. If we say C log P should be less than 5, that means we are putting the high quality compounds in that category. And that is why this comment is made, that high quality compounds require good solubility, so that C log P is below 5. And that can be obtained only if we do not invoke higher content of hydrophobicity in the molecule. Analysis of lot of data available from literature over a number of years has suggested some guidelines or has given some important information. Let us take a look at this slide. Let me divide the drugs available into the market into 4 categories. Class 1, where the membrane permeability is high and solubility is also high. This is the most preferred class. Class 2, where the high permeability and low solubility is observed. Class 3, low permeability and high solubility, and class 4, which is the least preferred, that is low permeability and low solubility. This is not preferred. Now, the data suggests that out of 41% marketed drugs, the new molecular entities, only 5% of the new molecular entities fall into the category of high permeability and high solubility. This is a point worth noticing, that only 5% of the new molecular entities, the newly synthesized drugs, only 5% fall into high permeability and high solubility category. Whereas 90%, 70% or plus 20%, 90% of the new molecular entities fall into the category of low permeability, low solubility or high permeability, low solubility. And solubility, you see, is the major concern over here. There is still a fraction 5% where you have permeability low, but solubility is sufficient. But the major problem which is faced by the industry is this 90% of the new molecular entities will either have high permeability but low solubility, or low permeability and low solubility, and that is the problem being faced by pharmaceutical industries. That is why I made that comment over there, that the high quality compounds means the solubility efforts, increasing the solubility, those efforts should be strengthened. Now, let us take a look at some more trends. Now, let us take a look at this data very carefully. This data corresponds to the affinity binding of the HIV protease inhibitors and the associated changes in standard free energy, standard enthalpy and standard entropy. Over a period of about 10 to 11 years, this data, it gives some interesting information. The green bars upwards represent enthalpy change, upwards we consider positive and downwards let us consider negative. Delta G naught is represented by the blue bars and minus T delta S is represented by the red bars. And the trend is for indinavir to dronavir. Dronavir in 2006, dronavir was the quite used FDA approved HIV protease inhibitor. Now, let us take a look at this equation. Delta G naught is equal to delta H naught minus T delta S naught. As I mentioned that one of the efforts in industry is to improve the affinity, and improving the affinity means that what should be done? Suppose if we want to improve the affinity by an order of 5. Now, we know that delta G naught is equal to minus R T log K, and if I want to improve the affinity by 10 raised to the power 5, that means by an order of 5, we can easily calculate that how much it will contribute to the value of delta G naught. This equation will permit that. However, delta G naught has to be negative. This has to be negative. And the contributions to delta G naught come from delta H naught and delta S naught, represented in this as minus T delta S naught. Reaction or the binding can be exothermic, can be endothermic. It can generate entropy, it can reduce entropy. The example that I would like to discuss with you gives some general trends observed over at least a period of 10 to 11 years. Let us take a look at the slide. 1995, look at the binding. The association was accompanied by positive change in enthalpy, endothermicity. And over the years, if you see, the green bars which were upwards are turning to downwards green bars. That means, over a period of about 10 to 11 years, the new molecules showed affinity with more exothermicity. See, it changed from endothermicity towards exothermicity, and this trend suggests that over this period, the new molecules have shown a trend changing from endothermicity to more exothermicity. Now, this figure, which represents the thermodynamic signature of all Hi1 protease inhibitors, suggests that enthalpies have accompanied the search for inhibitors with better binding affinity, selectivity and drug resistance profile. In pharmaceutical industry, we are not only concerned about the binding affinity, but the selectivity and the drug resistance profile also. Now, binding affinity, let us go back to this discussion. Binding affinity, affinity is talked in terms of delta G naught, and affinity can be engineered. Now, I am using the word very carefully, engineered. The affinity can be engineered in a molecule by either working on delta H naught or delta S naught. If we want to have a more negative value of delta G naught, delta H naught, if it is more negative, that will support, and if delta S naught also becomes more positive, that will also support. That means the binding affinity can be engineered either by making the interaction more exothermic or a combination of exothermicity and a lot of entropy generation. And let us discuss a bit more about that. The next comment, protease inhibitors with low picomolar affinity have favorable binding enthalpies. And that is what I was commenting upon, that picomolar affinity. You remember in one of the previous lectures I said that physical chemists would like to talk in terms of association and biochemists would like to talk in terms of dissociation. So, picomolar means we are talking here in terms of dissociation, and one can convert this dissociation constant into association constant and talks in terms of the binding affinity or binding constant. The trend is more clearly seen in this figure. This figure is on the statins. Statins are the cholesterol lowering drugs, and these act by inhibiting the HMD-CoA reductase, and therefore, the affinity of the statins to the enzyme becomes very important, and the efforts have been made to achieve the optimum affinity. Now, here if we look at the trend, once again the trend is towards more exothermic binding of the statins. Starting from fluvastatin to rosuvastatin, we see that the green bars, the length of the green bar is more, the extent of exothermicity is more when we started from earlier and made progress towards the later years. And of course, we should at the same time not forget the changes in the entropy, but engineering binding affinity in a molecule in terms of enthalpy becomes much more easier, as I will discuss later on. But a general comment which I can make over here is that from the first in class, this drug, if I say fluvastatin as the first in class in this period, to the best in class in this period I will call as rosuvastatin. So, from the first in class to the best in class, the trend has been enthalpic. That is, over the number of years, the new drug molecules were binding with a more exothermic character. Now, let us take a look at the comment. The thermodynamic signature of statins, the cholesterol lowering drugs inhibitors of HMG-CoA reductase have shown some general trend, that is from a lower exothermicity to more exothermicity in binding. And that is what is commented over here, that newer, more potent statins are enthalpically better optimized than older ones. This is enthalpically better optimized than this one. That is what the comment refers to. And as I mentioned, that when we want to engineer binding affinity in a drug molecule, we have to talk about the thermodynamic signature, which is shown in this figure. That means delta H naught and delta S naught are the two important contributions which will decide the sign and magnitude of delta G naught. So, what are the important contributions for engineering binding affinity? Now, let us say that this is the binding cavity. I represent this orange, this cavity as the binding cavity. And let us say we are interested that this molecule should go and fit into this cavity and bind over there. This is the process that we are interested in. So, what are the important contributions for this molecule to go and effectively bind over there? Let us remember that this molecule, when it is free in aqueous solution, it is surrounded by lot of water molecules, and therefore, before this molecule is removed from water and put into this binding cavity, water has to be removed from the immediate vicinity of this molecule, and that is what is called desolvation. This desolvation not only refers to the removal of water molecules from the vicinity of this drug molecule, but also the water molecules which may be occupying the binding cavity. Now, when this molecule is placed into this binding cavity, it will undergo some interactions with the constituents of this binding cavity. And what are those interactions? These can be hydrogen bonding interactions. It can be hydrophobic association also. There can be van der Waals changes, interactions, and there can be conformational change. Desolvation, hydrogen bonds, van der Waals interactions, conformational change, and if there are largely hydrophobic groups, there can be hydrophobic interactions also. Now, we need to go through it a little bit more carefully and closely. One is that the water molecules have to be removed from immediate vicinity of this molecule as well as from the binding site. And we talked about that once this molecule is placed over here, these interactions will set up. First is the desolvation. Desolvation is removal of water molecules, and these interactions, hydrogen bonding, van der Waals interactions, conformational change, or some hydrophobic interactions may also set up between the various groups of the drug molecule and the constituents of the binding cavity. And each point which is mentioned over here will contribute to delta G naught, delta H naught, and delta S naught. So, therefore, when we engineer binding affinity in a drug molecule, or we synthesize a new molecule with a good binding affinity, we need to take care of several things. One is the desolvation, how much energy it will cost to desolve it, how much energy it will give back when it forms hydrogen bond, van der Waals interactions. What will be the contribution to delta G naught or delta H naught because of hydrophobic interactions, and conformational change. Does it cost in terms of delta G naught or delta H naught, or does it give a reward in terms of delta G naught or delta H naught? Various thermodynamic quantities, when measured, give some guidelines in these directions in answering these questions. So, what we have discussed in this lecture is that over a sufficiently longer duration of time, the thermodynamic data gives sufficient guidelines towards the next step. For example, what we observed that the HIV-1 protease inhibitors, or we took the example of statins, the data suggested that the drug molecules from the first in class to the best in class have shown affinity which is more enthalpically driven. And similarly, the contribution of enthalpy or contribution of entropy, which makes changes to delta G naught, gives lot of guidelines, and these issues we will discuss in further detail in the next lecture. Thank you very much.